Okay, so think about this for a second. You have a blueprint so unbelievably detailed it can build an entire city, but it's so tiny it fits inside a microscopic speck. Well, that's your DNA, and it's tucked inside every single one of your trillions of cells. So the big question is, how does your body actually read that blueprint to build everything from your beating heart to the very thoughts in your head? Today, we're going to follow that incredible journey, right from that string of genetic code to a finished working protein. And this brings us right to the heart of the puzzle. You see, every cell in your body, with just a few exceptions, has the exact same master set of instructions. And yet, a liver cell does a totally different job than a skin cell. So how on earth does each cell know which part of that master plan to read, and just as importantly, which parts to ignore? That's the mystery we're diving into. All right, so here's our game plan. We're going to tackle this in 4 parts. First, we'll get a handle on what we're calling the blueprint problem. Then, we'll become molecular spies and watch the cell as it reads the genetic code. After that, we'll see how that message gets cleaned up in a process that's kind of like a cellular editing bay. And finally, we'll get to the really mind-blowing parts, how one single gene can end up creating hundreds of different proteins. So let's kick things off with that fundamental challenge. You've got this one single, massive instruction manual, but thousands of different, highly specialized workers that only need a page or 2 from it to do their job. How does that work? Well, this whole process of controlling which genes get turned on or off in a specific cell at a specific time, that's called regulating gene expression. Think of your DNA as a giant library of architectural plans. A cell that's building a bridge only needs the bridge plans, right? It couldn't care less about the skyscraper plans. So the cell has to be incredibly selective about which plans it photocopies and sends out to the construction site. Okay, so how does the build even get started? Well, the very first step is making that photocopy. In biology, we call this process transcription. This is where the cell creates a temporary working copy of a single gene from that DNA master blueprint. And the star of this particular show is an enzyme called RNA polymerase II. For our purposes, let's just call it the master scribe. Its whole job is to scoot along the DNA strand, read the genetic code, and write it down into a messenger molecule, a kind of rough draft, known as pre-messenger RNA. But here's where it gets really interesting. This master scribe, RNA polymerase II, isn't actually all that smart on its own. It's brilliant at copying, but it's a little lost. It needs a whole management team to tell it exactly where to start copying, when to start, and even how fast to work. These supervisors are a group of proteins we call transcription factors. And this management team, it's really split into 2 divisions. First, you have what are called basal factors. Think of them as the starter crew. They're absolutely essential just to get the process going at all. They get the scribe to the right starting line and get things moving at a kind of baseline, humming along level. But then you've got the regulatory factors. This is the real management team. They're the ones who fine-tune everything. They can tell the scribe to work way faster or slow down, or, you know what, just stop completely. So to put it simply, the basal factors turn the gene on, but the regulatory factors decide if the volume is a whisper or a full-on shout. And just to give you a sense of how unbelievably critical this fine-tuning is, get this. More than 20% of all the genes in the human genome are there for one reason and one reason only, to make these regulatory supervisor proteins. That is a massive chunk of our genetic real estate dedicated just to managing the rest of the blueprint. So how do these supervisors actually give their orders? Well, they interact with a control panel that's built right into the DNA itself. You've got the core promoter, which is basically the big red Start Here button. Nearby, you have these upstream promoter regions, which act like little volume knobs to tweak the level. But here's the part that's just wild. You also have these master switches called enhancers and silencers. These can be thousands of letters away on the DNA strand, but the DNA can actually loop around and fold over on itself, bringing these distant switches right up next to the gene to either crank its activity way up or shut it down completely. It's an amazing piece of molecular origami. Okay, so our scribe has made its copy, the pre-mRNA. But this thing is a rough draft. It's messy, it's got extra notes in the margins, it's not ready for prime time. Before it can be sent out to the factory floor, that's the ribosome, to actually build a protein, it needs to go through some serious quality control. You see, this first RNA copy isn't just messy, it's also really fragile. If the cell just sent it out of the nucleus as is, it would get chewed up and degraded in no time. So the cell has to proofread, protect, and properly package this message for its important journey. This whole process happens in 3 main steps. First up, a protective hard hat gets added to the front of the message. This gives it stability and helps the machinery know where to start reading. Second, a long shipping label, basically a tail of repeating A letters, is slapped on the back. And third, and this is the most dramatic part, is RNA splicing, where all the non-essential junk is literally snipped right out. So it turns out that the genetic message isn't just one long continuous instruction. It's actually broken up into pieces. The parts that contain the real protein recipe are called exons, but in between them are these long stretches of what seems like gibberish. We call those introns. Splicing is this incredibly precise process of cutting out all of those introns and then stitching the exons back together into one single coherent message. And this is definitely not some random cutting and pasting. There are specific little signals in the RNA code that act as cut here marks. Tiny molecular machines, called SNN-RNPs, recognize these signals. They all come together to form this huge, intricate editing complex called the spliceosome. And then, with the precision of a surgeon, the spliceosome snips out the intron and glues the exons together perfectly, every single time. And now, we get to what is, for my money, the most mind-blowing part of this whole story. Because this editing process, it isn't just about cleaning up the message. It's a source of incredible biological creativity. Right. So this is where the story takes a huge, fascinating turn. This editing bay isn't just about quality control. By choosing how to splice the message, the cell can create completely different sets of instructions from the exact same starting point. This is the real genius of the system. It's a process called alternative splicing. So imagine a gene has 3 useful parts, 3 exons. Let's call them A, B, and C. The cell could splice them all together to get ABC. But it could also decide to just skip exon B, creating a message that just reads AC. Now you have 2 totally different messages, which will create 2 totally different proteins that do different jobs, all from the exact same gene. And we're not talking about a minor tweak here and there. The scale of this is just staggering. Take one single gene in your inner ear that's critical for telling the difference between different musical notes. Through alternative splicing, that one gene can produce, are you ready for this? It can produce up to 576 different versions of the final protein. 576! That is how your ear gets its incredible sensitivity to pitch. So what does this all mean? It means this cellular editing bay is one of the main reasons that complex life, like you and me, can exist with a surprisingly small number of genes, only about 20,000. We don't need a separate gene for every single protein our body makes. Instead, we have this unbelievably elegant, efficient system for mixing and matching parts to create a massive library of tools from a limited set of blueprints. So, from a single master blueprint to a messy rough draft, through this sophisticated cellular editing bay, our bodies create this vast and dynamic library of proteins. It just goes to show you that complexity isn't always about having more parts. It's about the incredibly clever ways you use the parts you have. And it really makes you wonder, right? If this much creative power is hidden in just the editing process, what other secrets for creating complexity are still waiting for us to discover inside our cells?